We haven't solved world hunger, we haven't solved biodiversity crisis, we haven't solved climate change, we haven't solved anything, even the crisis in the agriculture, nothing has been solved, and the products that were promised just haven't come to materialize. And that wasn't because they didn't try. There was hardly a technology sector that had more money at its disposal than biotechnology in all forms and shades.
Welcome to Beyond the Fix. This is a podcast that takes a closer look at the solutions that science and technology aim to offer to today's most pressing problems. In this episode, we will dive into genetic engineering technologies that promise to tackle some of agriculture's biggest challenges: climate change, soil degradation, water scarcity, and emerging pests and diseases. Advocates argue that these technologies can help crops quickly adapt to climate stressors and enable sustainable agricultural intensification, so producing more on less land with fewer inputs. Today, we want to look beyond the fix and explore what merit these claims really hold. Can GMOs, so genetically modified organisms, deliver on the premises? And if so, what will be the implications on agriculture? I'm your host, Thomas Kobel, and I'm glad you're tuning in. My guest today is an agroecologist and entomologist with over 30 years of experience in studying genetically modified or genetically engineered plants and their impact on the environment. I think it's fair to say she has done some pioneering work in studying the ecology of GMO plants. Before she retired a few months ago, she has worked at the Institute for Integrative Biology at the ETH Zurich. Welcome to the show, Angelika Hilbeck.
Thank you very much for having me.
Angelika, you began your journey in this field back when it was still relatively new in the early 90s. Among other things, you have studied transgenic maize, engineered to be more resistant to a pest, the European corn borer, or Maiszünsler in German, which can seriously affect maize plants and lower yields. You showed that this genetically modified plant does not only adversely affect the target pest, the European corn borer, but also other non-target insects like lacewings and lady beetles. Can you tell us what you discovered about the GMO maize and its effects on these different target and non-target insects?
Okay, well, first of all, that maize, that GM maize I studied, expressed a toxin, an insecticidal toxin that was taken and engineered from a bacterium called Bacillus thuringiensis into maize, but also other crop plants like cotton, for example.
So this engineering there was to take something of a bacterium and put it into the maize, right?
Exactly. And it's not the entire bacterium. Sometimes people get that wrong. It's just the sequence of nucleotides that enable the plant to produce the toxin only, not the entire
so
bacteria.
That must be a certain protein? Or?
Yes, exactly. It's a protein. And fairly large protein, and it's insecticidal. And so the idea was the plant can defend itself against its herbivores, the pests, what we call pests. And this toxin has different, a varying spectrum of target pests. And the type of Bt toxin that was engineered into maize targets specifically lepidopteran pests like the European corn borer. That's specific. So the narrative was in those days that these toxins are so specific that they do not affect under no circumstances anything else other than that described target pest.
Okay, why is that? Because this bacterium also just targets these very specific...
Yeah, so okay. that would get us into the long evolution of Bacillus thuringiensis that has diversified into thousands, if not tens of thousands of different types of toxins targeting very different types of arthropods, insects. Some are specific for coleopteran pests, those are beetle pests. Some are specific for dipteran maggots. They're all targeting the juvenile stages of those organisms, not, or to a much lesser degree, the adult stages. So the target of these toxins are juvenile stages of specific classes of what we consider pest. Pest is an anthropogenic term. It's an
Of
herbivore
course.
that feeds, and if it feeds too much from our cropland, we call it a pest. But there's of course lots more herbivores on every plant. Yeah.
Okay, and so because they took a BT stands for Bacillus thuringiensis,
exactly.
that is targeted towards this very pest. They said, okay, it's only gonna affect this very species.
And it's not going to affect anything else. So I questioned that fairly soon, or right at, in the early 90s already, maybe because, or certainly because I did my PhD on Bacillus thuringiensis sprays, so formulated pesticides that are also used in organic production, for example. And so I knew a thing or the other about BT. And in the context of potato, that was in those days, and the potato beetle, the Colorado potato beetle. So I understood a little bit about the proclaimed and work mode of action of this, and what was engineered into GM crop plants was quite different in the number of key aspects that made me question that narrative that it will only and ever only affect the target pest. They changed. So, for example, because I based this narrative entirely on the knowledge that stemmed from using it as a formulated spray, which in those days definitely entailed the application of the toxins, including the spores. They were the products of fermentation processes, and it applied the proteins in an inactive form. It's a complicated thing. So what Bacillus thuringiensis it has a durable form of these proteins. It sits in the bacteria as a crystal form, and because it can take a while until a insect comes by and eats it, an insect must eat it, and it's a poison for the gut, for the intestines. So it must be ingested in order to unfold its effects. And only in the dietary tract, in the intestines, in the stomach, etc., then this crystal is dissolved.
Into the actual toxin, right?
Not yet. Into still inactive protoxin, it's called. And then you need to have, and for all of these things, you need to have the proper conditions in your stomach and the gut and the intestines in order to get the dissolution. It needs a very high pH of 10, 11, 12. And it needs the presence of a number of certain enzymes that then can cleave the protoxin, so reduce the size, basically cut it into half. And only that's then the activated toxin, what it's called, and that activated toxin can bind to certain epitopes on the gut of a susceptible insect. So it needs to have the right receptors in the gut, and then forms holes. It basically punches the gut, and that's what the insect then disintegrates and dies from.
Okay.
So you need the right pH, you need the right enzyme composition, you need to have the certain receptors, and it's a sequence of events that have to unfold before the BT toxin, the particular one, can actually exercise or unfold its effect.
And
if neither one is met, it's not going to do anything.
And that's the reason why you would think, oh, well, this is pretty targeted,
exactly.
Right? And
but and that's the big but that came to my mind immediately, is, okay, folks, what you've done in the plant cuts short this fairly long pathway of activation significantly and cuts out a number of steps that are necessary for specificity, for the proclaimed and known and verified specificity of them. So we don't need what's expressed in the plant is the activated toxin or near activated toxin.
Okay, so it has like skipped 2 steps actually. Okay,
exactly. So you don't need to have a certain pH anymore. That's done. You don't need to have the enzymes or only fewer of them, or maybe not even, depends. They are expressed in different forms even, yeah. So what is quintessential is what you need to have is the receptors. But we have the whole narrative of specificity was always based when you use the formulated or the crystalline forms. We have never studied which of these target pests only have the right receptors and could be affected if the toxin is presented to itself, but not the previous steps that are necessary. So it is conceivable. And what types of other insects may have the receptors, but when they swallowed the BT, they didn't have the proper pH and the proper enzyme composition to activate it, so it didn't do anything to them. So that whole narrative collapsed, in my view, of claiming it is specific based on the experience and the research that was done with full bacteria or formulated fermented products of bacteria, certainly crystalline products.
So this is where your research sort of started, right? To test this.
Exactly. And I just did my research and looked at, has anybody ever studied it? And of course not. And because it didn't exist, that possibility that you present the activated toxin to nature didn't exist prior to genetic engineering. Or in a much lesser amount, in a much more restricted way, within cadavers of affected caterpillars or so that died then.
Yes.
You know.
So, but why didn't they produce a mice that actually produces this crystal pre-toxin thing?
The protoxin?
Yeah.
I think it's a good question. I think this was beyond the technical capacity at that point. But also it was intentional because BT as a pesticide, as a formulated spray, was not liked very much by the farmers because it has a number of other issues as well. For example, it's very highly biodegradable. UV light disintegrates the whole thing or makes it inactive very quickly. Rain washes it off. So farmers don't like to use, certainly not conventional farmers. And in those days where I studied were conventional farmers who used BT sprays because none of the synthetic pesticides worked anymore on Colorado potato beetles. So they were desperate. They even used then, what's called for the organics. But they hated it. They hated it because you have a very small window of opportunity. On top of that, because you need to get the pest at a very early stage, only it works best on the smallest larval stages. So right after hatch from the egg, you need to catch it.
Okay, so
that leaves you a range of maybe 3 days.
Okay. So it was really intentional to engineer it that way.
And that overcomes all of these restrictions.
Yes. So maybe we can jump to the conclusions. Maybe not going to be a surprise.
Exactly. So what I did is what any thinking scientist, in my view, should have done in those days. But I was one of the first ones to actually get the money for it and do it, is let's see if the activated toxin either presented as a protein or presented through the plant in a kind of directly or indirectly via prey will also affect, for example, predators, larvae of important predatory biocontrol agents like lacewings or lady beetles. So I set up a study, not only one, it was pretty long studies, so many experiments, many replications, many tries, etc., etc. So it took many years. And sure enough, I found that you could kill chrysopid larvae to a significant extent, not like the target pest, obviously. Okay. So they weren't as susceptible as the target pest, but they were susceptible enough to die in a significantly larger proportions than in a control. And that kind of brought this whole very, very boldly and mindlessly pushed narrative in a dogmatic fashion by the industries that it doesn't and never will affect anything other than what we call a target pest. It collapsed it. So that made me immediately a target of the industries who did not like me to publish that.
To have exposed that, obviously.
Yes.
So you found out that also lady beetles, for instance.
Also lady beetles, lacewings.
And lacewings.
Yeah.
So for our German-speaking people in the audience, lady beetle, the Marienkäfer.
Right.
Lacewings are Florfliege. They look a bit like a green butterfly with sort of transparent wings, right?
They're beautiful.
They're beautiful, and also they are, you call them predators in German.
Predators, yes, because you differentiate between predators and parasitoids. Parasitoids parasitize a prey. Predators eat it, but they have very different types of eating. Feeding habits, etc. That also have to be taken into account.
So. Now it's a fascinating story, and I think it's also a very good example maybe to illustrate some more general processes that are going on when it comes to genetic engineering or GMO plants. So in the discussion with GMOs, you would say that there's maybe something oversimplified in the worldview or the mindset or the narrative behind GMOs. Can you maybe elaborate on that based on that example?
So the central dogma is something that's been debated since 50 years? No, longer, 70 almost. The idea to that and the DNA-centric way of perceiving how organisms work and how passing on of traits, of reproduction and evolution happens, is fundamental to genetic engineering. Okay, so it views, and that predates genetic engineering. It views DNA as a programmable code, and that's the entire language is borrowed from the IT sector. It starts with code. It goes on with programming cells and
biohacking, maybe.
Biohacking is just exactly the tip of the iceberg of the nonsense, basically. And continues to this day to other forms of genetic engineering that are called gene editing, genome editing. And what you do is you copy, you paste, you delete, you replace, the entire language is borrowed from the IT. And that's the image that's being conveyed with this language, is that cells, nuclei are basically nothing. Organisms are nothing else but programmable computers
and software.
Software we play up.
And so, why you call that nonsense? Why is it nonsense?
Because it's not true.
because
we know it's not true. So, and the argument, the scientific argument in the field, if you speak with geneticists or people, who work at the basic sciences, you find scientists, respected, known scientists, who will tell you that the whole concept of gene is nonsense. Mm-hmm. And it's because nobody ever saw genes. What you do is you associate with a certain sequence its nucleotide, its molecules, and a certain sequence of molecules gives you, with a certain confidence in statistics, a certain output under certain conditions. And then you say that's a gene, basically, okay? Mm-hmm. And they say that concept has already been misleading because, it's been taken to an extreme where the idea is being taught in every school, in every teaching book, is like that sequence of nucleotides is like a code.
Yes.
If you contain that, if you have that, you will do A, B, and C, and it doesn't matter where, and it doesn't matter who you are.
Yes, and also on the other hand, if there's a mistake in the code,
we fix it.
Or, I mean, you might be sick because, right. So, I mean, that's also the view I grew up with, obviously. Yeah. So can you maybe give me an example of why or how that is flawed?
Okay. All of these knowledges or these understandings are based on experiments. And I'm not saying that part of it is, I'm not saying that it's not totally true. Part of it may be true, but it is not the entire story. And when you look at the entire story, you start thinking about whether that part is even true. It's very complicated. Mm-hmm. So, for example, if you know certain diseases, you go to people who carry that disease, you study their genes, and you find statistically, you count genes, you count sequences of nucleotide sequences, okay? You count it and you're trying to find patterns in the sequence of nucleotide molecules in an organism, a person, an animal, a plant with a particular disease, and you screen a lot of them, and then you're trying to find certain patterns they have in common, certain expressions, certain nucleotide sequences. And when you find that, and you can find that, so I'm not denying that you cannot find that. Yes, you do. But then you associate with that finding, you associate causality. Mm-hmm. Causality that if you have that configuration of nucleotide sequences, you are more likely or close to 100% of expressing in your phenotype that disease. That said, there is diseases that almost 100% will happen if you carry certain sequences of nucleotides. But there's others, and there have been some studies done where people just went out randomly, not focusing on people with a certain phenotype, but randomly screening their nucleotide sequences and found that they also carry certain sequences, sometimes even homozygote, so on both alleles.
Yeah.
And they're healthy. They have never developed the disease. So very complex. And then, you go on and, maybe, then they started to, well, it was an uphill battle for those people who said, okay, DNA is not everything. They started to look at regulatory factors. They found what is now called epigenetics, for example, and said, okay, well, it's the regulation. Okay, that was sensational to finally find it. The people were long discredited until it finally, was like, okay, this is established knowledge. We have a layer above DNA that's regulated through RNA action that makes sure or determines which genes get switched on and off. But the way it's been configured now again in the tech field is, that's just another level we can engineer.
I mean, it sort of keeps, it already repairs the dogma somehow,
exactly.
Right?
It just adds layer of complexity to it.
Yes.
But there's others who say this is total nonsense. You're still giving to RNA and DNA, you're assigning a role of determinism to it that it doesn't have. Because it could as well just be, or in their view, and they have evidence to bolster that view, says it's not the DNA that determines what you are and what you can and cannot do, certainly when it comes to metabolic things, but it's a restraining condition. Mm-hmm. So you have a certain spectrum of possibilities that is kind of framed by what your DNA-RNA combination allows you to do. And it is then the environment that triggers reactions. And either you have an option to react to that or you don't. But which one gets activated is triggered by the environment. And the DNA just offers you certain possibilities to react to a certain cold, stress, whatever, what have you. If you have a suit that fits that condition, you can choose from, then you survive. If you don't, then you won't. So somebody was once saying it's more like a supermarket kind of thing, okay? It offers you possibilities. If you don't have the necessary tools in the supermarket, then you can't fix the problem. But it's not that the supermarket defines who you are.
Okay. So if I understand you correctly, what is, oversimplified or undercomplex in that view of, I just call it for now, biohacking, that you can just program genes like a software. I mean, also in the case of software, you sometimes have artifacts that you didn't intend. But so that the crucial role of environment is just not part of the equation.
Yes, it's not. It's totally not part of the equation.
And once you take that into consideration, what follows from that for the discussion of GMOs?
What follows from that is that, by and large, after 30 years of trying now, and actually gene technology, genetic engineering is 50 years old now. The first proof of concept that you can transfer nucleotide sequences into other organisms and get them to express to some degree in the lab was with famous experiments in California in the early 70s. 72, 73, somewhere there. So 74, 75 was the first conference where people started to think about whether there come risks along with it. But the proof of concept was done in the early 70s, so that's 50 years ago, half a century. And right from the get-go of why people pushed that technology so massively and so massive investments went into it was the prospect and the idea that you can engineer life at will. And that based on that dogmatic understanding of that genes are code, are the prime program that determines who you are and what you are. And you can now start to engineer at will with a human design and create new organisms. I mean, there was no limit to that. Still isn't, okay?
And there was also a lot of aversion to that idea, of course, because you would think of Frankenstein on steroids.
Exactly. So they kind of ignored that and were like, trust us, we only do what's good for people or something, But even if you take that dogmatic view of things at face value, 50 years, half a century down the road from when you remember what they started to offer and promise right from the get-go. Solving world hunger, you forgot to list that. That was a big, big, big one in the 90s. It went to the extreme that when you were against GMOs, you were for people starving to death. I mean, very, very polarized and very aggressive. So the sky was the limit to what you could do. That was the promise. Very bold, very grand. And when you look now, half a century later, basically none of it came to fruition. None of it manifested itself. The only thing, certainly in the agriculture sector, to this very day, since 30 years, is Bt crops and herbicide-tolerant crops. We haven't solved world hunger. We haven't solved biodiversity crisis. We haven't solved climate change. We haven't solved anything. Even the crisis in the agriculture, nothing has been solved. And the products that were promised just haven't come to materialize. And that wasn't because they didn't try. There was hardly a technology sector that had more money at its disposal than biotechnology in all forms and shades.
So what you are saying is it didn't happen because there's something wrong in the fundamental view that's behind it.
Work.
So in the last years, there has been a lot of talk about new genomic techniques, the most prominent being CRISPR-Cas. So because of that terminology, there's sort of the distinction between old and new genomic techniques. But would you say that when it comes to this fundamental flaw, this difference, how important is it?
There's no difference. It still builds on the same understanding of how genes in the minds of genetic engineers work. And it's been absolutely fascinating to observe over the decades how shut off and closed that circle is and shields itself to anything that might question the fundamental premise on which the technology rests. They don't hear it, they don't see it, they don't want to see it. It's become something like almost like a church, you know. It's a closed
belief
system, a self-referential belief system that shields itself to anything that doesn't fit their understanding, anything that questions the foundations, ignore even the foundations. And that's not a surprise because this technology has been taken to an extreme that you have primarily chemical engineers working there, biochemists, people who work at so small parts and in most reductionistic context possible. They are not interested in evolution of life. They are not doing basic science. They don't understand ecology. They have no clue.
So you wouldn't blame them for...
I don't blame them. I'm not an architect. I don't understand anything about architecture. I don't understand anything about car engineering or whatnot. That's okay. But what is not okay is to make bold claims for a field they don't understand anything about and insist that that's the only way to think about it in their terms and narratives and configurations, and ignoring and discrediting everybody who says there's maybe something wrong with your premise and you better go over the books and look hard because you're not delivering.
So I think this brings us to the field of politics and the political discourse, because this is, of course, not just a scientific question. And in the European Union right now, there's this discussion about deregulating GMOs. And in early 2024, there has been an open letter signed by 37 Nobel laureates and over 1,500 scientists. They've written this letter to members of the European Parliament asking them to deregulate. And there were also accusations in that letter that opposing new genomic techniques is an anti-science position. How do you react to that?
Well, that's a pretty poor argument. Huh? That is more revealing about those who write these than it does reveal about people who have valid criticism. As if you run out of arguments, you accuse the person of being anti-something, you know. So I think it's pretty revealing when it comes to that. It reveals more about the writers than it does about those they supposedly go against. And it's not the first time they're doing this. We had also letters written by Nobel laureates in the realm of golden rice once. Mm-hmm.
So golden rice was this also...
Vitamin A.
Vitamin A enriched GMO rice,
right,
supposed to prevent children from getting blind or even dying.
It's the same mindset behind. As I said, it's revealing more about the mindset of the people who write it than it does about those that they supposedly go against. I think for all these letters, it is the interesting part is when you look who hasn't signed.
What can you say about this?
And that's what I did for Golden Rice in those days when we saw the letters, and you know, which is situated in the context of world hunger and malnourishment.
Yes,
and it's people who have a very reductionistic, mechanistic understanding of what malnutrition and hunger is about, and they reduce it to be about a particular molecule you need to offer in a crop, and it's narrowed down to just productivity of a particular crop, like rice, for example, or maize. And if you fix that, then you at least spare the kids from going blind. And they pitch it as, you know, the only viable alternative there is, and it should be, well, they've mellowed it down. It used to be like the only ones are very absolute, and then it's like, oh, we need to use all possibilities, la la la. Today you don't hear that argument anymore, but it used to be very, very manifest. The problem with all of these things is it's people who don't understand the issue they're talking about. I'm sorry to say. They're not going for the causes. they're arguing for trying to treat symptoms. So if people starve, they don't starve because they wouldn't be able to produce food. And if they cannot produce food, don't look at the context why they can't. Hunger is always, without exception, situated in catastrophic context, either natural catastrophes, war, conflict, all of these things. If you're not addressing the causes of that, you're not solving the problem. And if you want to only solve vitamin A deficiency, for example, there is much cheaper, easier, proven ways of doing that by giving them vitamin A tablets. And that's done. And
But back to that open letter, you said you had a look at who didn't sign.
And so who didn't sign it was people who had knowledge and understanding of hunger. Economists, for example. Those are the people, especially there's some of them who have worked in the realm of developing countries and economics, in developing countries, in poverty-stricken areas, about poverty, access to not only food, but land, capital, etc. They have not signed.
So I want to mention a second point that was mentioned in that 2024 open letter, because there was the claim that the overwhelming, quote, overwhelming majority, end quote, of scientists supports GMOs. I mean, I don't know if that's true or not, but what do you make of that?
That's the same argument we heard 30 years ago. And,
Would you say it's true?
No, of course not. It's not true. Nobody speaks. We have millions of scientists out there. I mean, they've not gone around and did a participatory democratic opinion.
So the data
fight.
is weak,
Come on, there is no data. It's the gut feeling. Or it is their world. As I said, it's a shielded world these people live in. And they presume consensus in the gravitational area of their own club. So what makes up their club, that's the consensus, not understanding or being interested or ignoring that there is a larger community out there. It's like when you go to the Catholic Church and you ask them, they will always say, yes, God exists. But if you go out there, you may find a significant number of people who say, no, we don't think so. So, yes. And if you look at who signs it, it's people who have a common shared understanding of how the world works, have a shared ideology of how life works, the world works, politics works. And what they also all share is quite a self-confidence about themselves. But that comes with a Nobel Prize.
Well, I can't blame them if I had one. I don't know. But
I hope you would not lose.
Well, the chances are pretty small I will ever. So I want to come back to sort of the title of our podcast about the, behind the fixed. So what is fixed by GMO plants and by whom and for who, if we sort of look behind that fix?
Oh, what is fixed is for the developers and the companies to maintain their business model and design lock-in production systems where farmers are, stuck. So for them, that works. And what has happened is the business model has also shifted from what it used to be in the 90s. It used to be seeds.
Yes.
Okay. That's why the companies were in there. Seed companies. So the chemical companies mutated into seed companies, and now they're mutated into digital companies, and they have vertically integrated everything from one hand. What we see now is with the so-called genome editing, that particular method, or these methods, that's a group of methods you can apply to engineer organisms, has even delivered even less in the time since, you look at it, the official birth date is like 2012 when it was ready to be rolled out in a broad level. But of course, they've been developed and researched many years before already. So it's at least 13 years, let's say 15, if not more, that they're working on these so-called genome editing techniques. And we're now in the year 2024, going 25, we have on the market commercially available to farmers almost nothing. There were 2 tries, but none were CRISPR with a particular fatty acid altered soybean that crashed within a year or 2. We had a so-called an herbicide-tolerant oilseed rape, also not CRISPR, but ODM is the technology, the editing method, supposedly. They're not even sure about it, but that's what it was praised for many years. Also, that crashed. None of them exist anymore. They were on the market a few years. For the soybean, I think it wasn't even more than 1. They failed. These companies got out of the seed business, have merged now, and have explicitly said, we're not doing seeds anymore. We're doing traits. Traits means they're sequencing and pasting together certain nucleotide sequences they call genes and associate with a particular function, get it patented, and want to market traits.
Okay.
So that's the business model now. The focus has shifted from end products, from seeds, to the process with which you make money, because the entire process is patented. Whenever you use CRISPR, you pay license fees to those who invented it. Whenever you do certain types of manipulations, basically every step there is patented and you have to pay license fees. And that's thousands, tens of thousands per year, depending what you do it for and what's the application, etc. And the market envisioned product you get out of it. So what we're seeing is the shift to the process that the money is being made, which means you need to keep that promissory hype up even more so that people use the tools.
Okay, so now I start to see your point. So you said this narrative is sort of fundamental to this business model.
Absolutely.
That's why
it's even more fundamental to it, not less in any way or fashion, but it keeps it's startup companies these days. So you go to Silicon Valley, it's this entire disruption mentality of bringing out the latest, disruptive whatever in form of, you name it, whatever you think you can code in a petri dish. And you make the money primarily with the processes, the tools you need, the equipment, the consumables in it. All of it is patented, and that costs you a lot of money just trying and playing around with it. At the end, first of all, it's not. There's hardly anything out there,
yes.
and nothing
are you
useful.
saying this is a huge bubble that it's getting bigger and at some point it has to collapse?
Well,
also
maybe it does, yeah. What's called in the business world consolidation. We had a biotech bubble burst in the early 2000s already. Yeah. A lot of consolidation started then already, and I think this is going to probably happen. It depends how much people are willing to buy into this. Yeah. We've been seeing now, this summer was the first time, because we've been wondering, it's not only me who's saying that, there's a bunch of people who are following the business sector and are, looking at what's going on there. And we've been wondering, why is it that nobody asks the obvious question, where are the products? Yeah. You're not delivering. Well, one explanation is because much of that funding, because it's a startup culture, works with risk money. They pitch their business deals and their ideas to a community that has absolutely no understanding of the science behind it. It's hedge fund managers, it's, bankers, brokers,
venture
capitalists. And they can convince them because the theory, the narrative is very capturing.
Of course it is.
And it's laudable people who are behind it. It's massive science, Nobel Prize winners who are behind this. Of course, who you are, you're a broker, you're a hedge fund manager, you've got to manage a portfolio of what I call monopoly money, casino money, and and risk and failure is part of that. So they know they hedge their bets, they bet on certain proposals, and most of them fail. So that's part of that game. So it doesn't occur to them that they're not getting what they've been told. up until this summer now, we had from the business sector, not in the science field, in the business sector, a business enterprise who does analytics of business sectors came forward. Bonitas Research is that outfit, never heard of it before. They came out and looked at these two companies I just described earlier with the herbicide-tolerant canola and the fatty acid altered soybean that failed and then merged and are now in the trade business, etc., the trade business. They analyzed these companies, these two, and came with a damning verdict. It said they're misleading you.
Okay.
They're misleading you massively, and they're now collecting, based on that research and that evaluation that was damning, the law firms in the US, they have a very tough sword available, which we don't have. That's called class action lawsuit. Now there's at least half a dozen top-class law firms who are now collecting clients for launching class action lawsuits against these two companies that say you've just been trying after you failed and merged to get capital to pay out your early investors who put money into you and lost tremendous amount of money putting on you. You know, the stock prices were up way over $1,000. Now they're nothing. So
I see.
That may happen now.
Okay.
Finally, somebody
let's
bother to look. It's like, it's
see.
not happening. There's nothing out
It's
there.
very interesting also from an economic and sociological perspective and so on. But for the last few minutes, I would maybe sort of switch perspective a little bit and ask you, what alternatives to genetic engineering would you like to see flourish? Because the challenges, they are real, right? The climate change is real, hunger is real, and so on. So, because this is also, proponents sell this as, this is the quicker way, the more efficient way. And so maybe part of the narrative is we can't do it without GMOs because, this conventional stuff like breeding, it's not powerful enough. So but maybe you can say something about... Yes.
Well, the whole narrative of fast and efficient and so on, I think I've just debunked that.
Yes.
None of this was fast. There's nothing there. It's even slower than anything before. And it's not breeding. It's technical manipulation. So, but the alternative is, well, finally, how about addressing and remediating the causes of why things are bad in biodiversity, in climate change, and all these things? Why? So that's my favorite, okay? I'm old-fashioned. I like to go after the causes and remediate
and what would that mean?
That means you have to understand that the way we're using land these days, or since about almost 100 years, well, post-war, okay, we're 80 years, 70, 80 years, is when the chemical, that push started to its full extent and then up to today. That was a epic mistake. And people are not going to sign up to that. Some have, but, it was wrong in every aspect of it because it violated knowingly. Some say we didn't know better. That's not true. There were always people there who knew. They were just not listened to. Knowingly, they violated every rule that this planet and the ecological rule is based upon. They reduced diversity to monocultures. They replaced the loss of ecological functions that come along with using chemicals that destroy everything living in a field. Said, we don't need them. We replace them with chemicals. We don't need predators. We don't need parasitoids. It's all old-fashioned. We just kill what we want with synthetic pesticides. If plants are too weak, we fertilize them. Soils were considered substrate in those good old days. Nothing more than just hold a plant. And you could do away. We can do, as somebody recently put it, we can do nature better than nature. That attitude was really full-blown and unchallenged went through.
That's a nice way of putting it, but still, I don't think you're suggesting we just go back 100 years to where agriculture was like,
you can't,
with the horse on the field, etc. So what's the way to go now?
The way to go now, it will never be what it was 70 years ago. That's clear. There is no way back. There's only a way forward, but you have to go the way forward this time, understanding the processes the context we're embedded in, respect the natural boundaries that are given by nature to us, and operate within those boundaries. There is a term, planetary boundaries, for example, on some of those large, grand ecosystem processes. They have to be respected, and we have to bring and use what nature has to offer for us. Nature has everything at hand for us if we were willing to listen and to learn. Diversity is the best defense against shocks or against catastrophe. Redesigning diversity, bring diversity back, and, get away from killing substances. That's for sure. We cannot operate with deadly substances all the time and then be surprised that stuff is dead. That's the amazing part. People are like, oh no, all the insects are gone. Well, if you drench the landscape in insect-killing substances for half a century, they are gone.
Maybe one last question. I thought about, we could actually end on this note of, nature has everything. But how hopeful are you when you look into the future and the way forward?
Question. One
toughest to the end.
Smarter person than I am once said, and I wouldn't even know who said it, is it's way too late to be pessimistic. But there is, sometimes I am a little bit optimistic because I hear that at least today, you hear the European Commission to speak about the new Green Deal. You hear about them protecting, and doing policies that support and rejuvenate biodiversity, do something about climate change. But then again, so they have nice sounding policy goals and stuff in their texts. But if you go down and look at the detail, they have genetic engineering next to agroecology. They still want to only reduce. Well, I understand that these are processes that will take time. Mm-hmm. And I am willing to grant the time. And until a certain time, to transform systems like what needs to be done will take a decade or more to do. But it needs to be the common understanding that this is what we have to do. And everything you do from here to there has to serve the purpose of reaching that goal. So the goal must be to get out of pesticides entirely. They're killing substances. We need to do away with them. Full stop. And we can produce food without them. We can. But until then, you may have a phase of where you reduce it stepwise, etc. But they stop short of that. They say, okay, we want a reduction by 50% until 2030 or something, 45. And it sounds like that's the end point. Okay, if you say we want to reduce pesticides by 50% until 2030, irrespective of the fact that it's an illusion to be there in 5 years, basically, from now. And then what? Because 50% of using some of these pesticides is still horrible. It still kills all the networks of organisms and the ecosystem functions you need to support a agroecological functioning agriculture. You still kill it. You don't kill it half. There is no little bit of kill. That's not good enough.
So you would say we just have to do more.
We have to do more. I'm not even sure it has to be more. It has to be the right thing. And it doesn't come necessarily with foregoing something. The product is you will start seeing a diversity of foods that you haven't been seeing What's bad with that? You're getting products that have less residues of toxic chemicals in it. What's bad with that? What can possibly be the damage of that? On the way there, that's what you will start seeing. You will start seeing, hopefully, if it works, more beautiful insects and birds back. What's bad with that? But of course, farmers will say, but we need to make a living with it. Yes, we need to make a living with it. But that's a challenge that needs to be solved in the realm of economics.
And politics.
Yeah, and politics. But in the realm of ecology, we know what it takes. And we have enough systems ready to go. They need more research to be upscaled, outscaled, etc. But we know basically how to do that. We're not starting from scratch.
So I'll end on this.
Okay.
Finally, we found sort of hopeful notes. Thank you so much for being here and sharing all your knowledge. Thank you very much, Angelika.
Thank you.
In 2 weeks, we will talk with Wim Carton about negative emissions and overshoot. Basically, the idea that we can postpone decarbonization because we can just capture the excess carbon dioxide later. Beyond the Fix is brought to you by Critical Scientists Switzerland, an independent network of Swiss researchers, and is made possible by the Stiftung Mercator Schweiz. Music by Mich Gerber.